Well, hi there, molecular genetics fans. So we did our alignment analysis and have some, hopefully some better understandings about homologies in different organisms and looking at conservation of different gene products, proteins and things. So a really critical tool that we've been talking a little bit about, but I'd like to introduce and going to give you an exercise on, is the idea of the polymerase chain reaction. The polymerase chain reaction makes use of the thermostable DNA polymerase from Thermophilus aquaticus, known as TAC polymerase. And TAC polymerase has the ability to withstand very high boiling temperatures, and that in fact has an optimum activity at around 72 degrees. And it allows for the sequential denaturation of double-stranded DNA and then reannealing in the presence of a large amount of these little sequences that are called primers that flank the region of DNA that is to be amplified. So by looking at sequences, so you have to know a little bit of the DNA sequence, you can identify sequences that will serve as PCR primers and amplify up a region of a genome of interest. Another thing you can do is take primers from one organism and use them under basically low stringency conditions in another organism. So we'll be getting into that a little bit as well as we kind of progress through our discussion in sort of our in vitro lab. I'm going to be having you look at a gene in chicken and developing primers, and we're going to see if we can get it to work in other birds. I figure chickens, birds, they're all the same creature. They're all dinosaurs. Now, PCR has a number of rules and requirements and concepts that are important to think about. But by far the most important thing to understand with PCR is the development of these DNA sequences that we call primers. And what I'm going to do is provide you with an exercise in which you're going to be given a DNA sequence. A portion of the DNA sequence will be underlined. That's the portion to be amplified. And your job will be to develop PCR primers. So here, and along with this is going to be a sheet with rules and things, but just to discuss this a bit, here are some of the rules of developing good PCR primers. So a PCR primer, and no particular order, just as it's coming into my head, we're looking for it to be around 20 to 25 nucleotides is a good length. They can be shorter, they can be longer, but partly this is sort of my rules about how I develop PCR primers. And just as a little note, you can take a DNA sequence, and there's all kinds of software on the web now where you can put the sequence in and say, identify primers, and it'll bring them back to you. And that's not the point of this exercise. The point is to actually begin developing the understanding of how primers work. So we're going to do it by hand. So a 20 to 25 nucleotide piece of DNA is good. I'd say anything lower than 15, probably too small. Anything bigger than 25, and you really just don't, you're kind of just making nucleotides just to be making them. You don't need to do it any bigger. Because what we're looking for is a DNA sequence that will anneal, that will hybridize with a high accuracy, that it won't be hybridizing to random spots at a specific spot in the genome. And so because of that, we have a couple of things that we need to keep in mind. Another part of the criteria is ideally you don't want to have anything greater than a 3, maybe a 4 nucleotide repeats in this sequence. 3.4? No, 3 or 4 nucleotide repeats. You're trying to avoid those because those are not going to give you much specificity. Another thing we're going to think about is we like to have the GC content is approximately 50%. The other way of saying this is you don't want to be too AT rich. Having a higher GC content is probably not bad, but too much of an AT content is not great. This is going to affect, of course, the hybridization conditions, the annealing temperature, the TM. Remember, we've talked about this, that the greater the GC content, all things considered, the greater the TM. All right, so we want to have no 3 or 4 nucleotide repeats. We want to have, or I guess, nothing greater than 3 nucleotide repeats. A GC content about 50%. And again, if you can, we'd like for the last nucleotide, the 3' most nucleotide, would ideally be a G or a C. Part of this is because if we think about a piece of double-stranded DNA, or actually let's just go ahead and take just the top strand. So 5' to 3'. We're going to be hybridizing on this end because remember, DNA polymerization is always occurring from the 5' to 3' direction. And so we're going to have a primer that will hybridize here with the 5' end here, the 3' end here. And the Taq polymerase is going to recognize this 3' end and come in and begin putting in the next strand of DNA. And for the very first round of PCR, it just keeps on going. It goes as big as the genome is, and however long you let the enzyme process until you bring the temperature back up, you cycle the temperature back up. So because this 3' nucleotide is where the DNA polymerase is going to interact, if it's a G or a C, it tends to be bound a little bit tighter. Doesn't breathe and open up as much. So all things considered, if you can make the last nucleotide a G or a C, you're doing well. Now, PCR primers are a bit of a little bit of voodoo in that you can have a really good PCR primer and it may not work. And if that's the case, if you get either no PCR product or you get a lot of bands, it's not very specific, you change the primers a bit. You move over a few nucleotides either direction, you have to change the primer. Some primers work brilliantly. They're phenomenally clean all the time. Other primers are not. And that's just for the nature of this business. It's a bit complicated at the molecular level. All right, so now on to the really good parts. How do we develop a PCR primer? This is actually a really useful thing to think about because it really gets to the nitty-gritty of thinking about the relationship of these two strands. So let's just go ahead and throw out an arbitrary nucleotide sequence. GATTCATGCATGCCAATGG. Why not? That's just a sequence that just came to my head. Now, one thing is when I'm developing PCR primers for rather esoteric recombinant DNA experiments that I sometimes do, I'll have this whiteboard filled with nucleotides. And you can do it on the computer, but I actually like doing it on the whiteboard. So if it's just developing a straight PCR primer, yeah, I just go through the printed sheet or just mutter to myself on the screen and find it. But if you're having to add and subtract nucleotides, you're making changes in the nucleotides, I often find it really helpful to actually just write it all out. So here's the game. We have two PCR primers that are typically referred to as either the forward primer and reverse primer. So the forward primer and the reverse primer. Another way to think of this is the left side primer and the right side primer. So here is the game then. What we're going to do is with a double-stranded molecule, and we're going to just take this little chunk here. That's just double-stranded here. GAATTC, 5' end to 3' end. We have to develop a primer that'll hybridize to this nucleotide sequence such that it will generate a 3' end going in the direction that we want to go. So the lower strand is going to be this, CTTAAG 3' to 5'. All right, so in this case, what we're going to do is, for the forward primer, we denature the DNA at the denaturation phase of the polymerase chain reaction. We denature the DNA by boiling it, and we are left with single-stranded DNA. So if this is our primer, it's going to hybridize to this portion of the genome, this portion of the sequence. And there's our 3' end, and when we add the DNA polymerase, we're off to the races. So the point of this is the forward primer, or the left side primer, is easy to interpret. It is simply the nucleotide sequence. So the forward primer, that's easy. All you do is you look at the DNA sequence, scan around it, find a spot you like, and write that nucleotide sequence down and send that to the company, make me a nucleotide, boom, you're done. So the forward is simply the top strand, 5' to 3'. Now, the reverse primer, or the right side primer, that one requires a little bit of deconvoluting, kind of standing on your head. Because when you're given sequence, you're always given sequence that represents the top strand. We have to develop a primer that's going to hybridize to the bottom strand. Now, we have to develop a primer that's going to hybridize to that top strand. It's going to be related to the sequence to the bottom strand. So let's go ahead now and erase our forward primer. And I'll tell you that this is, conceptually not trivial. It does take a little bit of thinking, and it's a little bit like standing on your head writing in a mirror. But I've got some rules that if you follow, it just works. So there you go. All right, let's go ahead and complement this sequence. So before we do this, here's the rule for developing the right side primer. You scan along the DNA sequence, you find the region that you want to use as your primer, that is flanking the downstream right side of the sequence you want to amplify. And the first thing you do, so this is the development of the reverse primer. First, you find the sequence that you like. All right, so you identify the sequence that you like. Next, you write it as the complement. Or you complement the sequence. You're a lovely sequence. So let's go ahead and complement a piece of this. It's going to be CTTAAG TAACGGTAACCTAACC. All right, so that's the bottom strand. Now remember, when we're given sequence, we're only given the top strand. The bottom strand is inferred. You just use your base pair knowledge and you make the bottom strand. The bottom strand is running in the opposite orientation. It's running in the 5' to 3' direction. So, number one is you identify the sequence. Number two, you complement it. That's the sequence. Now in the complement. Now here's the trick. Oligonucleotide synthesizers can only build DNA in the 5' to 3' direction. So in order to have the sequence presented to the oligonucleotide sequencer, you have to present it in the 5' to 3' direction, which means 3, you have to now write the sequence in reverse. you have to write it backwards. So that becomes the following. CCA TTGGC AATGGCAT GAATTC. All right? That now is in the 5' 3' direction, and it represents the complement to the top strand. So those are the 3 critical rules for developing the reverse primer. You first identify the sequence, you write it the complement, and then you write it in reverse. So I'm going to be giving you an exercise in which you will do exactly this. All right, now generally when I'm making PCR primers, that's where I stop. One of the things you can do, and it's actually a very useful sort of experiment, is to take these sequences and blast them. And blast will automatically know, okay, this is a short sequence, and it'll adjust its parameters. And it will scan through the genome that you're looking for. And you can assign, say, I'm going to be looking at the Pseudomonas bacterium. I want to screen only the Pseudomonas bacterium, not all the genomes that have been sequenced and all the DNA sequences, just the Pseudomonas genome. And I want to identify, are there other spots in the genome that this primer might hybridize to? Because if there's a bunch, then you might go, not the best primer. You could try it and see what happens. One of the things is if you get a primer that hybridizes here, but you don't have the complement other primer hybridizing on the other side, you're not going to get an exponential increase in reaction. You're not going to generate the actual large amounts of DNA that the polymerase chain reaction can generate. And so you'll end up with background. It'll be a bit of a mess, but it might still work. What you want to make sure, though, is if you have too many spots that are hybridizing near each other, that could be a problem. So it's a useful tool. Usually what I do is identify the primer and go, this is the best I can do, screw it, make the primer, and if it works, it does. If it doesn't, I go, oh, rats, I probably should have scanned or remake the primer. All right, so let's just go back through for a second, though, and see what the primer, how it influences the polymerase chain reaction. And I got to be honest with you, in molecular genetics, or in molecular cell, we covered the polymerase chain reaction and did our own PCR experiment actually right before we had to break for our spring break that is still going on. So we've covered this in molecular cell, but just to refresh you, the polymerase chain reaction operates in a series of cycles, and you've got a set, within each cycle, a set of temperature changes that are doing some really important things. So we start out, if this is going to be our temperature axis, and this is going to be time. We'll go ahead and do this in seconds, and we'll do temp in degrees centigrade. We always start out our PCR reaction at around 95 degrees, and in fact this is sort of canned into the programs on most PCR machines, is that this high temperature is what you start out at. This stage of the reaction, of the polymerase chain reaction, is known as, of course, denaturation. And at 95 degrees, there is no double-strand DNA that can withstand at 95 degrees. Everybody is going to denature. The hydrogen bonds break. So we typically denature, for somewhere around 30 seconds. Often, sort of some of the stuff that is done in PCR is almost like folklore. The very first round of denaturation is often done for 2 to 3 minutes, sometimes 5 minutes, mainly because you're now working with genomic DNA entirely, and so it takes a while to completely denature it, fine. But then once you actually start the cycles, the denaturation time is a good bit shorter. 30 seconds is usually pretty good. Next, we drop down in temperature, and let's say we'll drop down to 53 degrees centigrade. I just invented that. This is going to be the temperature that will allow the primers to anneal. And this is known, of course, as the annealing stage, the annealing phase. The annealing temperature is usually the TM minus 10 degrees. That's a good place to start. Now, when you're making PCR primers, the manufacturer will give you what the TM is, or there's websites that'll do it for you. And you would ideally like to have two primers that have fairly similar TMs. But if one primer has a bit higher GC content than the other, that's why we try and shoot for 50%. And all these things are rules. You do the best you can. If one primer is 50% GC and one primer is 70% GC, the 70% primer is going to have a higher TM. And you have to be 10 degrees below the lower TM. And this is one of those things you can begin to adjust. If you've got a really good primer set that works well, you might actually be able to have a very high specificity, no background, at like 5 degrees below the TM. If, on the other hand, often what I do is I have a primer from one organism, I'm going to try and use that to amplify a DNA sequence in another organism, I might drop the TM, it might drop below more than 10 degrees, I might drop 15 degrees, maybe even 20 degrees below the TM. That allows for base pair mismatch. All right, so that has to be pretty much just determined empirically. That's the annealing temperature. And again, annealing is anywhere from 30 seconds to maybe 1 minute. Part of it is we have this huge molar excess of primers, and so we really don't need to have a lot of time floating around. The amount of primer is in way excess to the amount of template of DNA you put in, and so it hybridizes pretty darn quickly. Next, we raise the temperature up to, I'm going to mark on this side, nothing back here, 72 degrees. 72 degrees, this is the optimal temperature for Taq DNA polymerase. This is known as the extension phase of the polymerase chain reaction. So the extension phase. And the extension phase time generally is determined by the length of the PCR product that you're generating. The usual rule of thumb is probably about 1,000 nucleotides per minute. You know, what this means is that the Taq DNA polymerase doesn't instantly zip out a piece of DNA. It takes some time. So if you're doing a short fragment, like a couple hundred nucleotides, which is often, PCR generally is less than 1,000 nucleotides, but you can go for 30 seconds to a minute. If you're doing a really long fragment, and in our chicken experiment we may be doing a couple of kb, you may actually have the extension phase going for 2 to 3 to 5 minutes. So then the extension phase, whatever time it's going to be, we'll just bring that over here. And now we go back to denaturation, and this is one PCR cycle. So generally, a PCR program will run 30 cycles, 25 cycles. Sometimes they'll run 42, 45 cycles. The more cycles you do, you can potentially get more DNA, but you can also start to introduce more background. The fewer cycles you do, the more accurate the material you're generating is, but you don't have a whole lot you can see. And when we do PCR and we run the products out in a gel, this is referred to as endpoint PCR. And for endpoint PCR, we have to have generated enough DNA that we can visualize it on a band. We can visualize the band on a gel. Now, that means that you probably have to run 30, 35 cycles. Doing real-time PCR, you actually are measuring the fluorescence of the product that you're generating. And the fluorescence can be detected at a much, much lower concentration than seen on the band. So you can detect PCR product using a real-time PCR machine at a much lower point. We'll talk about real-time PCR later in the semester, but real-time PCR is typically what's being used right now to do COVID-19 tests, in part because the analysis is done while the PCR is running and you don't have to run a gel. But for our purposes, for around here, we typically run gels to look at our PCR product. Also, we use PCR to generate pieces of DNA that we're actually going to clone. We actually will take that piece of DNA out of a gel, cut it out, the PCR product, and use it. So, from our purposes, we usually run about 35 cycles or so. All right, so that's the PCR reaction. We'll be getting into some genome organization and genome evolution a bit. It's some of the stuff that's interesting, but not all that exciting in terms of molecular genetics. We'll be kind of plowing through that in the next week or so pretty fast. Then to get to some interesting things in terms of gene expression and stuff like that. So that's the exercise I have for you. Give it a shot. In terms of it being due, it'll be due next week, and I'll be looking at your alignment assignments. They look good so far. I think everybody's doing really well. Thank you for your support and participation. Come by and see me if you have any questions. Don't be a stranger. And I'm out. Peace.